I have with me today Professor Kurt Buchmann from University of Copenhagen, and we are talking about the special issues of International Journal of Parasitology, which focused on fish parasites. Kurt contributed one of the papers to the journal, and as you know, the journal was going to be published and coinciding with the 10th International Symposium on Fish Parasites, which unfortunately had to be postponed to next year because of COVID-19. So hopefully we'll meet next year at the ISFP 10. But Kurt, you are sort of like a veteran of ISFP conferences. I think you are someone who's been to almost all of them. Do you have any favorite memories or anything you would like to tell us about the previous ISFP conferences?
Oh yes, they have always been great conferences, actually. I have never seen any bad conference at all within this framework, and it has always been extremely stimulating. So, well, I didn't attend the first meeting, but from the second one, I was present, except for 1 year where I was actually organizing another conference just at the same time. So I was the local organizer here, and therefore I couldn't join the meeting in the Czech Republic, which was actually organized a little later than ours, but they coincided. So unfortunately, I couldn't attend there. But many of the good colleagues actually passed by Copenhagen as well during the same week. So that was great. Well, anyway, I think that the International Symposium for Fish Parasitology, these conferences, they have always been so stimulating. And maybe I should mention that the original idea was to make the East and West collaborate, because they all started during the Cold War, where we had the Iron Curtain through Europe. And everybody knew that our Eastern colleagues, they made extremely good research, and they had been doing that. They had a great tradition for many decades and hundreds of years within fish parasitology. And the same went for the other side of the curtain in the West. So people took the initiative to make these two groups collaborate. And I think the first one was in the Czech Republic, in Czechoslovakia at that time, and the second one was then in Tshirt in Hungary, and that was where I started my career within this business.
So was it your first trip to communist country?
Yes, I think so. Yes, that was my first time to a communist country, but it was quite stimulating. It went to the nice little town called Tshirt, located near Lake Balaton in Hungary, and we had a great time there.
So it sounds like an excellent networking between the scientists from the Eastern Europe and the West. Do you have any photos you could share with us?
I just found a few photos I had in my collection. It was about starting up the collaboration between East and West. And, these days, I still think we have the need to do that again, actually, because we are setting up new curtains in the world these days. So I think that the future, and maybe it should start up in Australia, let's hope so, in 21. But yeah, but at least here I went to Tshirt in Hungary in 87, and later on the next meeting was in Petrozavodsk in Russia in 91, and later on in 95 it was in Munich in Germany, and then in the Czech Republic 99, and then we went to South Africa, Bloemfontein 2003 I think it was, and then Italy, Viterbo, organized by Simonetta Mattiucci, I remember. And then we went to Viña del Mar in Chile, and the latest
Marcela
one.
Oliva organized this one.
Yeah, exactly. Yeah, it was a great meeting as well. Excellent. And then finally to Valencia in Spain in 15. And then of course it was the plan that the next one should be in Australia, and suddenly we were standing here with this pandemic situation. Yes.
Well, I still hope it will happen next year. It looks like the conferences are really great for not just networking, but also sightseeing.
Now I switch to the next one here. Actually, you may remember at the Tihanye meeting. It was located just by the Lake Balaton in Hungary. And one day we had a social tour, a little boat trip on the lake. And I took a photo of my good old friend, the late Mr. Gyrodactylus salaris, Jørgen Malmberg. He described that monogenian in 1957. And he's sitting here on the boat. And we also had some other renowned fish parasitologists which were writing the literature at that time. That was Clive Kennedy here at the left, and in the middle, Telavo Valtonen from Jyväskylä in Finland, and Jan Thulin here from Sweden. And on the same boat, you may see here the Professor Horst Harchevsky from Karlsruhe, now in Germany. He has always been the expert in fish acanthocephalans and nematodes, of course. And here next to him, the Viking Ville Hemmingsen from Tromsø in Norway when he was young and fresh. So, but that was a great time there, and we all enjoyed it, and we started a lot of collaboration, which is still ongoing now these days, actually. So that's a wonderful thing to look back here and see the old colleagues. And then the next meeting was in Petrozavodsk in Russia, and here you see a nice photo of the group. And that time, well, there probably were many fish parasitologists around, but the meetings were not that big, as you can see here.
Yeah.
But especially the good old man here, Dr. Oleg Bauer, was leading the Zoological Institute of that time, or Albina Gajewskaya from Crimea here. So there were many of the well-known parasitologists around here. And when I write up here that we had the meeting in the midst of the political inferno, it was because of that time the Mr. Gorbachev, he was actually introducing perestroika, we had the impression that not all people in the Soviet Union were agreeing with him. And actually during the last day of the conference, he was actually put in, taken into custody. And that means that the entire group here was isolated because they cut down all the communication lines. So my good colleagues here, we were actually, they were actually stuck in Petrozavodsk without any connection to families or anything for 1 week. And but the funny thing was that this is my supervisor, Marianne Koy here. Here we have the Csaba Székely from Hungary. But Marianne and we actually left in the morning when they took Gorbachev. And we traveled by train to St. Petersburg, and we got out with the first airplane there. But the rest of the crew here, they were actually, I think they were left here for 6 days before they actually could go on. Yeah. And the rest of what happened with Mr. Gorbachev, you can read in the history books, of course, but he is still alive, and he still has an opinion of what is going on over in Russia. Then just a little photo from South Africa in Bloemfontein, because here we have Kalman Molnar, the well-known and renowned fish parasitologist, and we are standing there watching lions on the savannah in Africa here. So that was also quite an experience. But it was really fascinating to be together with the African fish parasitologist here. It was really nice to have this collaboration, actually. And then the next meeting was in Italy, in Viterbo. And here you can see the crew has grown quite a lot since we were standing in Petrozavodsk. I will not mention all of them, but you may recognize both Australians and Europeans and Americans, so and people from the Eastern Europe. So that was also a great meeting and a wonderful atmosphere in Viterbo that year.
Yeah, it was
And
fantastic place also, like a
yeah.
really nice place. Yeah.
And then there was this Vineyard del Mar, and then we were ending up in Valencia, and you can really see that the group has grown quite a lot. So let's see if we have any airplane companies left when we get to the meeting in Australia. I do not know how many we can be there. Okay, but that. So I'm looking forward to Australia. Yeah. Hopefully.
We're also looking forward to seeing everybody in Australia. And I don't bring any pictures, but I have a T-shirt I wanted to show you. I don't know if you remember this T-shirt.
Yeah, lovely.
This is from the conference in Viterbo. So
Yes.
organized, and I think we were given all these T-shirts, and I still have mine, and it's been really good. So the conferences are really fantastic, aren't they? So really good. And as you said, you develop collaborations with people from all over the world, and you continue working with them after the symposium.
Yes.
So you obviously go to these meetings to talk about fish parasites. So I just wanted to ask you a little bit about which parasites do you think are the most important for fish, particularly commercial fish, maybe in Denmark and Europe in general?
Well, I have been with fish parasites for and really been enjoying fish parasites for more than 40 years. So I would not take one single species and say that this is the most fantastic one. They are all wonderful. They are part of the life. So and according to my view, the parasites are the most important animals in this world. I mean, I think that maybe more than 75% of all species are actually parasites. It's the most successful way of life. But I would say that when we talk about commercial fisheries and so on, we have one important example from the Baltic Sea these days, and that was actually what our little article in the International Journal for Parasitology was dealing with. The Baltic Sea is a semi-enclosed marine area, actually a brackish water area, and it is quite young and unstable. Because it appeared actually less than 10,000 years ago after the glacial age, where the ice melted, the ice cap melted away from the area. And first we had a freshwater lake called the Ankylos Lake. Later on, salt water moved in from the Atlantic Ocean via parts of Sweden, and then it became a marine area. And with that, we had introduction of cod. We had introduction of salmon, Atlantic salmon, and we also had seals coming in and spread, herring, and a number of other species. But after that period, actually, these fish species were trapped in the Baltic Sea because the way the passage through Sweden was closed again, because Sweden was elevating up after the pressure was lifted from the ice. And then we had a brackish water system with all these marine species, and the only salt water coming in was through a canal through where we have Denmark these days. And nowadays we have the Great Belt, Little Belt, and the Sound moving salt water into this area. So these fish species, they are living isolated together with seals in this little community. And it is fascinating that it is so unstable. If one species actually gets into trouble, it will affect the other species quite fast, because they are not having long range migration, for example, as we see by Greenland or Iceland or the Faroe Island or other places in the Pacific, where the fish can migrate out of problems. Here in the Baltic Sea, we have seen that the gray seals, which they have been there together with the fish for many thousands years, yes. But we have probably seen these fluctuations in the populations, and what I think is that we have this fluctuations of the populations because the seals, the large top predator, lives in the same area with the fish they eat, and at the same time they spread their parasite there. They have roundworms, which are anisakid roundworms called the Contrasecum osculatum, and to a minor degree the Pseudoterranova decipiens. And especially the Contrasecum osculatum search-stage larvae affect the cod physiology so strongly that we have seen a tremendous decrease of the cod population. When I was studying these Baltic cod around 35, 40 years ago, the total stock was about 700,000 tons, and these days it's less than 200,000 tons. So 500,000 tons of the standing stock has disappeared, and that is probably partly due to predation from the seals. But of course, when the parasites, which are adult in the stomach of the seal, will infect the cod and weaken them extremely. They are much easier to catch for the seal, and this predation and maybe also other factors have affected the population of the cod. So now it's so low that you don't believe it. But we have studied the progress of infection, because during the 1980s, I actually investigated thousands and thousands of Baltic cod, and we rarely saw this parasite because at that time we didn't have many seals. Maybe we only had about, at least less than 1,000 seals in the entire Baltic Sea. And now we have more than 40,000 of them, and they are populating all areas of the Baltic Sea and spreading the parasites to the Baltic cod. So that is this development has been in a way fascinating, but also quite disturbing for the cod fisheries. So it has had extremely high importance for the fisheries there, if we should drag one parasite out here. But Barbara, we are dealing a lot with with other problems with fish. We are mostly dealing with aquaculture, and rainbow trout is actually one of the most important species we are dealing with. We do a lot of research on the different bacterial diseases and parasitic diseases of a rainbow trout, and recently we have been looking into not only vaccination and other prophylactic ways of keeping this fish at good health, but we are also doing some genetic breeding these days, where we are doing SNP analysis in order to find the genotypes which actually will confer a good disease-resistant phenotype to the fish. And recently we published about one little parasite called Ictiophthirius multifilius, or as somebody say, ick. It's easier to say ick. This is a devastating parasite. Actually, it can kill off maybe almost 100% of the population if you don't interfere. But it seems that we found that the genes conferring some resistance to this parasite, they are located on chromosome number 16 and 17 of the rainbow trout. And I really think that it is fascinating to dig into this way of preventing disease in farms. I mean, you can do it by fast breeding. You do the SNP analysis of the fish, and then you use this information to go to the breeders and select the breeders very fast, and then you make a new generation with a much better resistance towards the disease. I think that is one of the future ways we should enhance aquaculture business in the world, actually, by doing this. Vaccination is extremely important, of course, yes, but also when we are going to vaccinate fish, we have to make the vaccine. We have to have some a crew coming into the system vaccinating the fish or maybe we should invest in a machine vaccinating the fish. But all these things, they are expensive. And in addition to this, we have to starve the fish before we vaccinate them, and they are starved after vaccination. And when each day we starve a fish, we have a quite a big production loss. So if we can actually just breed disease-resistant fish, I think that's the way forward. And at least we are just contributing a little with studying this parasite Ichthyophthirus multifilius, which are affecting most freshwater species in the world. And if we can use this technology in the future, I think that that is something we should discuss also at the future meeting in Australia. I'm looking forward to that.
Yeah, definitely. It's very interesting, the disease resistance and breeding disease resistant fish. I agree with you. It's one of the way forward. And also because there are no commercial vaccines available against parasites in general, and it's really hard to develop one. So I think it's the more practical even to look for genetically resistant stock. So you've been going through you said you've been working as a parasitologist for over 40 years, and I'm quite amazed at the depth of what you are doing, because you talked a bit about taxonomy, a bit about ecology, a bit about managing the diseases and solving the problems through genetics. So you really learn different disciplines as you were trying to solve problems with parasites. Is it how it progressed?
Well, I think it's just a matter of love to life, actually. I think that it's beautiful to live in this world, even if we have a number of problems, actually, we know that. But I think that it is so fascinating just to be present in this world. And I know that we are not living here forever. But anyway, to enjoy life, I would say that is to work with parasites and to study them and find their secrets.
So you obviously have had a very distinguished scientific career, but as far as I know, you also like painting. So do you paint parasites or do you paint just fish? Because I've seen quite a few pictures of fish that you've painted.
Yeah, I paint a lot of parasites. I have to admit that, but I'm not professional. That's also because I just love parasites. I paint them.
So do you like painting parasites as a relaxation, or do you do it to share your knowledge of parasites, show other people what parasites look like?
First of all, I have to admit that I can't help it. I just have to paint them. I mean, it's just a feeling coming out from my guts, then I paint them. But to be honest, I also use them when I do my teaching, actually, in fish diseases. I also paint bacteria and virus and different kinds of parasites, because I think that the visual effect is also important in the pedagogical process. And even I know that I'm not Van Gogh or any fancy painter. I think even a bad painting by Kurt Buchmann can help understanding the basis of parasitology. So that's why I'm using them also for teaching.
So do you consider your painting scientific illustration or art?
Well, I'm a modest man, so I just say illustrations. It must be illustrations, and maybe in 200 years people will talk about art in 2020. But for the moment, I'm just doing the illustrations, right?
I really enjoyed seeing them. I remember your PhD students often use your pictures for the cover of their thesis, and that's how I remember that you were painting fish and parasites. So this is really fascinating to listen to all this, and I'm really sorry we couldn't meet this year in Cairns, but I hope we'll meet next year in Cairns in July. And I would like to congratulate you on your publication in IJP, and looking forward to the next paper, which is going to be on disease resistance to parasites. And that's a really fascinating area, and I hope to hear more about it next year. Thank you very much for your time.
Thank you. Thank you for calling me. It was a pleasure talking with you again, Barbara. Thanks a lot. And see you soon, at least. in Australia. But I also hope to see you before. Thank you very much, and bye-bye.
Bye.